Today we will discuss about the cestodes. So under cestode, the human as a definitive host. So when we consider the definitive host as a human, Tinea solium, Tinea saginata, Hymelepis diminuta and Hymelepis nana. In these four species, human act as a definitive host. So in Sri Lanka, they reported all five species: Tinea solium, Tinea saginata, Hymelepis diminuta, and Hymelepis nana, and the Echinococcus granulosus. So in cases man act as a intermediate host in Echinococcus granulosus and the Tinea solium. So in here the man act as a definitive host and intermediate host in Tinea solium. So according to the infective stage or the stages present in the human, the human act as a definitive host or the intermediate host. So if the human ingested the larval stage, so this larval stage become adult, so that time man act as a definitive host. Sometimes in the case of Tinea solium, man get infection through ingestion of the egg, or sometimes accidentally if they infected, that time the egg become a larva. So this stage, in this step, the human act as a intermediate host. So human as an accidental host. So in the case of Bertilla studii, the shape, when we consider the shape of the cestode, it is flat. That means generally they are flattened. It's look like a ribbon shape. So you can see the picture here. This one, it is look like ribbon. So so the location adult in a small intestine of the vertebrate. So where the habitat of this parasite or location, the adults, they are present in the small intestine of the vertebrate. Size, when we consider the size of this parasite, it is few millimeter to the meter, right, is in size. So other one, a plant or animal that has reproductive organs normally associated with both male and female sex. So in here, when we consider the reproductive system, both male and female present in the single segment or organism. So here we cannot separate male and female. So it is endoparasite. Endoparasite means it is present inside the host. So elongated flat body containing head, scolex, strobili, immature, mature, and gravid proglottids. So when we consider the body, it is flattened. We already discussed, dorso-ventrally flattened, and it's contain anterior part, the head we call especially here scolex, and then it's continuing the strobili, here we call the segments. So when we consider the next part of the scolex, we call immature segment, then it become mature segment, then it become gravid proglottids. So when we consider the scolex or head part, So here organ of attachment but not associated with the intake of food. So the main function of the scolex is attachment, attached to the wall of the intestine, right, intestinal wall in host cells. So but they cannot take the foods by using these scolex. So function to maintain the position of the tapeworm in the small intestine. So when we consider the small intestine, when we take food or water, so it come through the small intestine. So if the parasite is present, normally what happen, when we take water, it's come with the water, but to prevent this one, they use this colex, especially these suckers or scolex, that mean the hooklets. By using that, they are attached to the small intestine wall. So they cannot move, right. So it contains 4 suckers on the scolex, and some species we can observe the rostellum. Usually hooks are arranged in one or more circular anterior to the sucker. So here when we consider the hooks, they are arranged in 1 rows or 2 rows, and we can see the suckers around the body, the head part. So tapeworm sometimes it's appear as a armed or unarmed tapeworm. So if the tapeworm contain rostellum, so we call armed tapeworm, so armed head or armed scolex. If the tapeworm does not contain the rostellum, we call unarmed tapeworm. So here this is the Tinea saginata, Tinea Sodium scolex. So there are 5 species we all know. In the case of Tinea Solium and the Tinea saginata. First picture, Tinea saginata. Here there is no rostellum and the hooklets, right. Only we can observe the suckers, it is appear pear shape. But in the case of Tinea Solium head, it is contain the hooklet, and this is the rostellum, right. This is the part, the rostellum. This is rostellum, and surrounding the rostellum we can see the 4 suckers, the 2 rows of hooklets, and 4 suckers we can observe. So other thing, Echinococcus granulosus also similar morphology of the scolex, that means similar to Tinea Solium. Then when we consider the Hymelopis diminuta and Hymelopis nana, normally compare with Hymelopis species, Tinea species is very important because the Tinea Solium is very dangerous compared with even Tinea saginata or Hymelopis. So, and other thing, when we consider the Tinea Solium and saginata, this is larger compared with Hymelopis species. So here we can see the rostellum in diminuta, but here we cannot see the hooks, right. And here also we can observe the 4 suckers, and in the case of Hymelopis nana, we can see 4 suckers and short rostellum with hooks, 1 rows of hooks. So these are the morphological features of scolex of Cestodes. So you have to remember this. So the commonly we can see suckers in all 4 or all 5 species. Rostellum hooklets, it may present or not present. Right, rostellum and hooklets not present in only Tinea saginata. Actually, rostellum, right? Rostellum not present in Tinea saginata, but rostellum present in all other 4 species: Tinea solium, Hymelops diminuta, Hymelops nana, Echinococcus granulosus. In the case of hooks, Tinea saginata and the Hymelops diminuta does not contain the hooks. So only Tinea solium, Echinococcus granulosus and the Hymelops nana which are contain the hooks. Okay, clear? Then when we consider Hymelops nana, which is a single row hooks, but in the case of Tinea solium and the Echinococcus, 2 rows of hooks. Right, now we will move the neck part, germinal or growth region. Actually, after head part, the neck is present. It is undifferentiated shown, contain stem cells that are responsible for giving rise to new proglottids. So head, near the head part, the end of the head part, actually end of the head part, we can see the neck. This is undifferentiated zone. We cannot differentiate cells or the segments wise. So it is stem cell. That means stem cell mean it can spontaneously divide, grow and divide and differentiated. So this stem cell grow, and if it is, when it is matured, it is produce the proglottids. It is immature, then it become mature, then it become more mature. We call gravid proglottids. Stobulation mean this is differentiation of the new proglottids near the anterior end. So then if we can differentiate the proglottids or segments, we call stobulation. So stobula or body we call the segments actually. So arise from the neck. So stobula mean from neck to end part we call the end of the segment, last segment we call stobula. This is arise from the neck. So consist of reproductive organ of both sexes. So when we consider stobula, each contain each and every segment contain both sex, male and female. Each set is referred to as a genitellum. Right here, when we consider A, B, C, this is the head part. So head part, and after end of the head, we can see the neck part. From neck part, we call all together stobulations, right? Stobula. So this is immature one. So B is the immature one, mature segment. C is the mature segment. D is the gravid proglottids, that is fully matured. Here we can the segment D, we can see the egg and the internal part very well compared with other. So consist of 3 parts. So stobula we can divide into 3 parts. Immature proglottids, this is very youngest segment. Then mature proglottids, so mature segment locate between immature and gravid proglottids. So contain male and female reproductive organs which are completely matured and capable of reproduction. So they are the completely mature proglottids. So mature segment, they are completely matured. It's contain male and female. So here you can see the this one. the reproductive organs, right? Then gravid proglodids, they fill up with the eggs. So after the reproduction, the gravid proglodids is fulfilled with the eggs. So here you can see the actual the tapeworm. So this is the scolex, right? This is scolex. Here you can see this is head part. Actually immediately you can see the head part. Then we call this all the ribbon like things we call stabila. So here you can see here this one. In here you can see this area actually immature one. Here you can see the mature one, this one. This area you can see the mature one. So here you can see here inside the magnifying, when it is magnifying, you can see the testis and uterus and ovary, and here you can see axil gland. So here you can see the both male and female reproductive organs, right? Then after that, end of this the ribbon, this is anterior part. The posterior part we can see the gravid proglodids, right? This is the magnifying one. So you can see here the gravid proglodids filled with the egg, and we can see the uterine branches, right? And other thing, when we consider the scolex, here you can see suckers, 4 suckers, and hooks. If it is present, hook you can see. Other one, this is the rostellum. This is the rostellum, right? This is the rostellum. And end of the head part, we can see the neck, right? This also same thing. Here you can see anterior part, head. You can see here the upper view. There are 4 suckers. Rostellum, this is the rostellum. This is hooklets. This is suckers, and here you can see the neck. Then and the posterior part, this is the posterior part. This is posterior part. Here you can see the reproductive structure, that means fully matured part we call gravid proglodids. But when we consider the life cycle of the cestode, this is the common life cycle. So attached to the small intestine, begin to produce the stabile. So first where we started from, right? Here we will start from here. Here, this is the starting point. Definitive host becomes infected by the ingesting the intermediate host containing the larval stage. So here definitive host. So according to the species, definitive host may vary. It may be human or the dog. So if the definitive host infected by ingesting the intermediate host containing the larva. So when the definitive host, when they take the larva containing intermediate host part, right? So that intermediate host contain larva. These larva enter into the definitive host. So in here, so these after entering into the definitive host, juvenile mean the very young tapeworm will develop, and this is attached to the small intestine wall by using suckers and the hooks, then it produce the strobili. These strobili, that means segment. This segment become immature segment, then it become mature segment, then it become a gravid proglottids, right? So it is present inside the definitive host of the small intestine. So these gravid proglottids pass to the outside environment. So then when it is fully matured, sometimes these gravid proglottids remove from the segments, right, strobili. So this is passed with the stool to the environment, outside environment. So the proglottids ruptured and the release embryo or egg. So after the release from the strobili segments, these gravid proglottids, it is passed with the stool to the outside environment, normal environment. So this is in environment, these proglottids ruptured and which proglottids containing eggs, large number of egg, and it will release these egg or sometimes embryo ingested by intermediate host. Then it is developed into larval stage inside the intermediate host. Then these infected intermediate host part ingested by definitive host, it will continue the life cycle. This is common life cycle. So here in this life cycle, the larval stage is the infective stage, right? But in some cases, the intermediate host, the infective stage is sometimes egg also. So here the comparison of Taenia solium and the Taenia saginata. So here you can see first rostellum in Taenia solium, the head part. When we consider the head part or the scolex, in Taenia saginata only we can see the suckers, but in Taenia solium we can see suckers plus rostellum with hooks, right? This is the specific characteristic of Taenia solium scolex part. Then when we consider the mature proglottids, it is appear like case of Taenia saginata, there are so many lateral branches. That means when we consider right side or left side, it is more than 15. Here more than 15 lateral branches, but here 7 to 15 lateral branches. That means less than 15 branches, right? And other thing, when we consider the egg morphology, we cannot differentiate the egg. In the case of Taenia saginata and the Taenia solium, the egg morphologies are similar, and these egg become a cysticercus larvae. So in the case of Taenia saginata, cysticercus bovis, the name, larval name is Cysticercus bovis. But in the case of this one, the Taenia solium, Cysticercus cellulosiae. Cellulosiae, right? So then this Cysticercus cellulosiae become an adult, right? This is the life cycle. And morphological differences of Taenia solium and the Taenia saginata. Then when we consider the Taenia solium, we call also fork tapeworm. The distribution prevalence in countries where the fork is eaten uncooked or partially cooked. So here where we can see see the Tinea sodium where the fork is eaten with uncooked. That means they are taking raw and then without cooking or partially cooking or undercook or half cooking. In that area we can see the Tinea sodium. So in the case of Sri Lanka, few cases reported. So when we consider the Adulthinia Sodium, the habitat or location in main small intestine. When we consider the length of the Tinea Sodium, it is 3 to 5 meters. So this is very larger. When we consider the scolex or head part, it is bare or it is containing a rostellum and has a 4 suckers with a double row of hooks, and other thing rostellum with 2 rows of hooks, and we can see 4 suckers and rostellum here the arm shape, right, with 2 rows of hooks. Then when we consider the egg, and we can see the striated wall and embryo inside we call oncosphere. So we will discuss during the egg morphology, so don't worry. The larval stage is here we call Cysticercus cellulosae. So if they ask infective stage or regarding larval stage, so you have to write full answer Cysticercus cellulosae, which are found in skeletal or cardiac muscle of the pig. So pig is the intermediate host. So here when we consider cardiac muscle or skeletal muscles, in these areas we can see the larva, the Cysticercus cellulosae. So if they ask what is the infective stage of Tinea solium, which are produced teneasis, so you have to tell how, or sometimes how the men get the tinea, the infection. So you have to tell ingestion of undercooked or uncooked the Cysticercus cellulosae larva containing pig, right? So you have to write full answer. So here when we consider the Tinea solium, the width is 1 millimeter, the scolex, then this is, so you have to remember this thing, right? This is 2 to 8 meter, and when we consider the segment, it is 80 to 1000 segment in Tinea solium. Right, and when we consider the gravid proglottids, it contains 7 to 12 uterine branches on each side, right? Both sides we can count more than 7 and less than 12, right? And here we can see A and B, first picture and second picture from left side. Here the first one, the brownish color one, this is the liver biopsy. Here you can see the whitish color, that is the larva, that means Cysticercus cellulosae larvae. So this is B, this is the after taking the Cysticercus cellulosae, and this is after removing of the Cysticercus cellulosae. This is the appearance. So third picture, brownish color, you can see this is the egg of the Tinea solium, and fourth one, here you can see the dark brown color here, this one. Dark brown color is the outer shell thin membrane. This is dark brown color, but inner membrane means striated one, this area, right? This area we can see, this is the area we call inner cell, this is the radially striated, right? So this is the radially striated the cell and the oncosphere together we call embryopore. Right? Remember, embryopore mean oncosphere, that means inside the part, and the striated cell. Right? When we consider Cysticercus lava, actually it's appear this picture, right? This is appear, we call this is bladder, and here this scolex this is actually immature head part. It contain the lava contain immature head part. So in the case of Taenia, so in the case of Taenia solium, Taenia saginata and Echinococcus granulosus, the egg we cannot differentiate but morphologically similar. So here carefully observe this one. They start from here, first one here. The most outside part we call thin cell membrane, right? Very thin. Then second one, this layer is striated embryopore. That means embryopore mean the striated part and the oncosphere. This is oncosphere, third one. Inside the oncosphere we can see the 3 pairs of hooklets. So up to these two we call embryopore, right? Remember this. That means oncosphere plus the striated inner part we call embryopore. Right, here again you can see the morphological feature. First one, the head part. Second, B is all the full of the Taenia solium, right? This is full body length, first one. Second one, this is the head part, and other part is in immature segment. This is third one, mature segment. Fourth one, gravid proglottid. This is separated part, right? Separated single segment, right? Right, here you can see the mature proglottids of Taenia solium and saginata. This is Taenia saginata. This is Taenia solium, because here you can see in first picture, this one, first one, it is single segmented. Here you can see double, more than one segment. So again it is indicate the species differences, and you can count the lateral branches. These are the lateral branches, so you can count. So in this picture, fourth and fifth, third and fourth, here you can see lateral branches in Taenia saginata, and each side more than 15, here less than 13, right, each side. Right, when we consider the life cycle of Taenia solium occur in the small intestine of the human, so life cycle continue in small intestine of the human. Human is the definitive host. So in the case of Taenia solium, human act as both definitive host and intermediate host. Humans are infected by eating raw or undercooked infected food and the contaminated food, water, hand to mouth. So this is the mode of transmission. How human get this infection, that means Taenia solium. Main thing, they are infected by the eating raw meat without cooking, Undercooked mean partially cooked, half boiled, half cooked. Infected larva containing fork, right? Or otherwise we can other mode of transmission contaminated food, water, or hand to mouth. So once the egg hatch, the larva penetrate the intestine and enter the bloodstream and may develop into cysticerci, bladder worm. That's why we call the bladder worm in the muscles, skeletal cardiac muscle and the brain and eye, right? So this is the life cycle of the parasite. So first we started from ingestion, right? Definitive host. So when a human take raw or uncooked infected fork. So I start from here. This is the first step. When they take the fork here, this if larva is present, this larva come and attach to the small intestine, and it become, after attachment it started to segmentize, so it will grow. That means it will grow. So and then we can if infected person shaded the segments or egg when they passing the stool sample. So this is stool sample taken by, that means the infective stage, the segments or the egg taken by the fork, right? This is the life cycle. So the two clinical form of the disease in the case of Taenia solium, here clinically it's produced two disease condition. One is teniasis. This is due to adult tapeworm. So if when the person, the human, when they take the raw or undercooked fork, so which containing the Cysticercus cellulosae larvae. So when human take this one, so these larvae become adult. So these adult produce the clinical symptoms or clinical features or clinical disease we call teniasis. In this situation, human act as a definitive host. Other situation, sometimes here egg may be the infective stage. So here infective stage is larva. But in cysticercosis, next disease, we call the infective stage is egg, right? Remember this. So egg. So here what happened after ingestion or accidentally when we take the egg, this egg become a larva. So here human act as a intermediate host. So the disease condition we call cysticercosis. So when we consider the teniasis and the cysticercosis is the very dangerous, because it is the larval stage lodged into the nerve system, CNS, central nervous system, and it produce neurocysticercosis. It's lead to death. So compare with teniasis, cysticercosis very danger. Or compare with Taenia saginata, Taenia solium is the most dangerous one. Right, clinical feature, teniasis due to adult years. Symptoms are rare. Actually infected person, they are not showing any specific symptoms. Proglotid pass in the stool. So even they don't have any symptoms, but the patient they will pass the proglottids. But cysticercosis, this is due to Cysticercus cellulose larvae. So cysticercosis occur when the Taenia solium eggs are accidentally ingested by the human. So here accidentally humans are take the eggs, Taenia solium. So as a medical laboratory scientist or medical students, so you should handle the sample if when you are suspected as a Taenia species. So if egg is present, it accidentally you may infect. So when you are handling the sample, you have to wear safety measures. So eggs in contaminated food, water, or sometimes egg may present in the water or foods. Sometimes auto-infection, egg from anus, mouth, head and mouth. So vomiting of eggs so segmented by the infected person, these are the possible how the cysticercosis occur, right? Clinical features of cysticercosis depending on the location of cyst. So the larval stage where it is present, the clinical features also vary. So if it is affect the nervous system, cerebral cysticercosis or neurocysticercosis. If it is affect the eye, so ocular cysticercosis. If it is affect the muscles, muscle cysticercosis. If it is present in heart tissues, heart and cardiac cysticercosis. The prevention and control, neurocysticercosis has a large impact on the human health because neurocysticercosis is produced by the larval stage. That means when human take the egg, it become a larva. So this is this larva lodge into the central nervous system. It is produced neurocysticercosis. The Taenia solium is the only source of infection in human and the pig. So when we consider these, so humans are the humans and the pig are the main sources, right? So epidemiologically controllable and Taenia solium can be controlled with a few and simple steps. So we all know human and the pigs are the only source. So by considering these two, we can control that. So the cook pork product etiquette. So we have to cook in a high temperature, then only internal part of the fork meat will kill the or destroy the larval stage. The patient with Taenia solium infection should be treated immediately and fecal matter disposed of cautiously. So if any patient identified as a Taenia solium positive, we have to immediately treat the patient because actually that is the cysticercosis, especially if they identify, immediately we have to treat, and if they pass the stool sample, we have to carefully dispose it. Chances of auto-infection or ingestion of egg must be limited. So when we handle the sample, so we have to consider carefully, we have to use the safety precautions because there is a auto-infection possibilities. So the stool of food handler of the recent immigrant from the endemic countries should be tested for the proglodids. Stool of the food handler. So in some cases recently the endemic area, if any person visited to the endemic area mean where the normally very high the disease present in high level, high concentration. If any person visited that area, so we have to, especially if they are the food handlers, so we have to check right, for the proglodids. And public awareness and the personal hygiene practices and proper meat inspection. So these are the possible way how to prevent the disease, the Tinea solium. Then Tinea saginata we call beef tapeworm. So the common in countries where the beef is eaten raw or under cook. So here where we can see the beef tapeworm or saginata in where beef are taken much raw or under same like fork. Here also if we take raw or undercooked meat, half boiled or partially boiled, when you are taking like that, so you can get the infection if the beef is infected. Few cases reported in Sri Lanka, so Tinea saginata also reported in Sri Lanka, usually 3 to 5 meter in length, so can grow up to 20 meter length. So here when we consider the Tinea solium and saginata, Tinea solium length is smaller, that means shorter than Tinea solium is shorter, Tinea saginata longer. Right, the scolex does not have a rostellum, but in the case of Tinea solium they contain rostellum at the scolex, but here in the case of saginata there is no rostellum, but here we can see 4 suckers in the scolex. Same the features we can see in Tinea solium also. In Tinea solium also we can see the 4 suckers and highly extended stobula, that means stobula the length, when we consider the length of the stobula, so it is extended than the Tinea solium. But the Tinea saginata is the largest of the genus Tinea. So here this is the largest one when we consider Tinea genus. So in the under genus Tinea there are so many species and this genus we are learning Tinea solium and Tinea saginata, but when we consider all Tinea genus, Tinea saginata species is the larger one, right, can survive in many years. Another thing is the Tinea saginata, they can survive many years. And the mature proglodids contain a uterus, ovary, genital pore, testis and vitelline gland. It does not have the digestive system, no mouth and no anus. So here in the case of Tinea species we can see the uterus branches, ovary, genital pore, testis. So these are the things we can observe in proglodids, mean mature proglodids, and yeah, it's actually mature proglodids. So here man is the definitive host because adult worm we can see in the man, cattle or beef is the intermediate host. So here in the case of Tinea saginata or beef tapeworm, the infective stage is the larval stage, right? So Cestodes bovis larvae. So when you are taking ingestion or eating of raw or uncooked infected beef, the infected larva, that means at the bracket you have to tell the name Cestodes bovis larva containing beef. When we are eating raw or undercooked infected larva contain beef, right? This is the Tinea. Egg of the Tinea saginata are not infective to humans. So here you have to remember this is the one of the MCQ point. So in the case of Taenia solium, the egg also infective, larva also infective, but in the case of Taenia saginata, only larva is the infective form, egg is not infective. So this is the morphology of the different part Taenia saginata. First picture, it describes the Taenia saginata's collex. It is 1 to 2 millimeter, because we already discussed Taenia saginata is larger than the Taenia solium, but in Taenia solium's collex, the width when we consider 1 millimeter, here 1 to 2 millimeter, little bit larger. And same like Taenia solium, here we can see the 4 suckers, but the other difference